Hey guys, Kenna here. Today we're going to go ahead and go over some basic genetics and Punnett squares. Now, for some of you, this may be very familiar. You probably did some of this in your middle school years. For others, it may not, and that's okay. So that's why we're going to go ahead and go over it here, is so that we're ready to go ahead and use Punnett squares and understand the basics of terms like dominant and recessive. Okay? So let's go ahead and start out with our essential questions. Number one, how is genetic information, forms of a gene, carried from one organism to its offspring? Now, we've talked a little bit about the process of meiosis and how we go ahead and create our sex cells or gametes. Remembering, hopefully, that we get one copy of each chromosome in each one of our sex cells. So you get one allele, not two. Number two, where does an organism get its unique characteristics? Okay. When we start thinking about this, this is that process of going from our DNA, going through transcription, translation, making the amino acid sequence that folds into the protein, which gives you the actual trait. We're going to try and tie all this together with some very important vocabulary today so that we have a better understanding of these kind of basic genetics and what processes are responsible for life's unity and diversity. Then we're going to go ahead and show you the Punnett Square. And for most of you, once I show it to you, you'll be like, oh, I remember going over this. But we need to make sure that you understand the Punnett Square and how to use it so that we can go ahead and do crosses as we continue on through genetics. Okay, so let's get started. First thing I want to go over is the genome. Now, we've talked about this a couple of times, but this essentially is all of the genes together. Now, the average mammal, including humans, probably has somewhere between 30 and 60,000 genes comprising his genome. That's a number that's down from what we originally thought that humans had over 100,000 genes. But after completing the Human Genome Project, we discovered that that just wasn't actually the case. In eukaryotes, the genome is found within the chromosomes. The number and conformation of these chromosomes is specific to the particular species. So human chromosomes are different from, say, goldfish chromosomes. We have 46. Goldfish have something like 94. So that number does not necessarily mean more complex or more highly evolved. It simply is a difference in the number of genes per chromosome and the size of the chromosomes. Now, some terms that we introduced you to when we were going through meiosis were diploid and haploid. Now, you have to remember that a body cell is going to be diploid. It has two sets. But when we go ahead and take a look at a sex cell, sperm or egg, they're going to be haploid and have just one set of chromosomes. Now, one set of chromosomes carries one copy of the genome. Hence, diploid organisms, like you and me, have two sets of chromosomes, one from our mothers and one from our fathers, okay, maternal and paternal, to make a set. So each set carrying the same gene, loci. Loci is the position, though not necessarily the same alleles, at each locus. Okay, so these underlined words here are some key vocabulary that you should definitely know. So locus is the position, loci being its plural, and you can only have one allele per chromosome at a particular locus. The two chromosomes in a given organism, remember the maternal and the paternal, that have exactly the same array of gene loci are said to be homologous. This is that homologous pair we talked about when we were going over meiosis. When dealing with gene locus in a diploid organism, an individual can be either homozygous, homo means same, or heterozygous, meaning different. So a homozygous individual is going to have two of the same chromosome, allele, I should say. Okay. So homozygous, two of the same allele. Okay. Which is different when we look at heterozygous, two different alleles. One too many L's. Okay. So you get the idea. Let's talk a little bit more about genes and inheritance. Now, Genes contain the information that's going to allow us to produce the proteins. We've talked a lot over last term and this term already about the fact that genes themselves are instructions. They don't do that much. Between the genes, we're going to go ahead and call this the genotype. Okay, genotype is the collection of the instructions to make the trait. That can be found on your chromosomes. And let's go ahead and zoom in on one of these little bands here. And this is going to represent a single gene. Now, that gene then is going to go through protein synthesis, transcription, translation, and it's going to go ahead and produce the protein. Now, remember, proteins are responsible for the physical expression of the gene as phenotypic characteristics. So this is what we call the phenotype. So we think about something like eye color, hair color, skin color. These are phenotypes, and they are caused by the protein that's coming from that particular gene. Now, in eyes, there may be more than just a positive for this or that. And when we start thinking about dominance and recessiveness, we can look at eye color, and it's a little more complicated than some of the other genes. But the idea is that the trait itself comes from the manufacture of proteins. Those proteins are manufactured because of protein synthesis for a particular gene. So the proteins have a huge role in determining the trait of the organism. Since you inherit your genes from your parents, you inherit your traits for your parents. It's not a one-to-one, but that's the general rule. It depends on this idea of dominance and recessiveness. Now, we are focusing here in this particular example and in most of what we're going to cover on sexual reproduction. In organisms that reproduce sexually, which means using the sperm and the egg, we're going to inherit traits from the gametes. Remember, the gametes are the sperm and egg, okay, or pollen and ova, depending on if you're doing plants or animals. And these are produced by the process known as meiosis, which is a form of reduction division. Offspring then are going to possess a combination of genes from both the parents, maternal and paternal, depending on how dominant a particular trait is. This is not necessarily true with asexual reproduction, because in asexual reproduction, there's not this mixing of the traits of mom and dad. It's an exact copy of the original DNA, barring some mutation. There is no meiosis. We're simply going to do mitosis to produce an identical clone. Now, there are a few exceptions where we start seeing some exchange of alleles or DNA material from clones that live in colonies, but this is not the normal. Okay. One example of this that's actually a really important one is the development of things like antibiotic resistance in bacteria. This can be transferred from one bacteria to another via a copied version of a DNA plasmid, but we'll leave that alone for now. And as I mentioned, we're going to focus on sexual reproduction. Now, most plants and animals reproduce this way, at least part of their life cycle. This involves genetic input from two parents, so a mother and a father, and the random assortment of what gene you get from what parent provides new combinations in your offspring. Except for identical twins, the offspring are all going to be unique in their gene combinations. The diversity in genetic makeup of offspring means that some may be better able to survive and reproduce in the particular environment they live in. This will be huge when we go ahead and start talking about evolution later on. This idea of survival of the fittest, or survival and reproduction. In sexually reproducing organisms, favorable mutations can combine in a single individual and give them an advantage in a particular environment, making them more likely to survive and have more offspring. Okay? This is part of why when you go ahead and take a look at families, you typically can see some resemblance between the generations, the grandparents, to the parents, to the children, and so on and so forth, because they do share some genetic instructions, which are going to go ahead and produce similar proteins, which are going to give them similar traits. Now, this one I put in a handout that's attached to the notes, okay? So you can look at that, get that post. Sexual reproduction has adaptive advantages. If you live in a changing environment, you have a lot of variety, and those individuals who have the best combination of traits are going to be more likely to survive and pass on those traits. But if the environment changes, there's enough variety to go ahead and find new, better traits that fit for that particular environment. Okay, that's different with asexual reproduction because asexual reproduction is allowing them to capitalize on a particular environment, but it doesn't necessarily allow them to adapt or change well if the environment changes. And these clones that we see from asexual reproduction are much more vulnerable to changes in the environment. Okay, you can produce a lot more quickly using asexual reproduction, but it doesn't give you the variation that makes sexual reproduction more favorable in changing world. Okay. Let's talk a little bit about the terminology with respect to locations of genes. You'll notice on this particular example, you have a homologous pair with on the left being the chromosome from the egg or the maternal chromosome, and the one on the right being the chromosome from the sperm or the paternal organism. The position of a gene, whether it's gene A or gene B, is the same on both chromosomes because we're looking at the same chromosome. However, the allele for that chromosome is not the same. The egg gives a recessive lowercase a, the sperm gives an uppercase dominant a. But notice that they're at the same position, and remember we call that position the locus. Okay? In sexually reproducing organisms, when the sperm and the egg come together, you end up with two of the same chromosome or a homologous pair, one from each parent. If you look down at B, gene B has a particular locus that is not the same as gene A. This particular trait, we're looking at the chromosome from the egg has the dominant version, and the allele from the sperm is the recessive version. Notice that you do not have to have all dominant or all recessive on a particular chromosome. Okay. Here's the locus for gene A. Here's the locus for gene B. When we go ahead and take a look at this, you have to notice that the two different genes have different alleles at that particular loci, but they're for the same trait at the same position on the same chromosome. All right. Now, when we go ahead and start thinking about identifying these traits, this diagram is going to illustrate the complete complement of a hypothetical organism. So it's totally made up. It has a total of 10 chromosomes, 5 nearly identical pairs. So you can go ahead and start looking at them, and you look at the banding patterns. For example, here's one, here's the other one, here's two, here's the other two, three, and so on and so forth. So we line up these homologous pairs, one from dad called the paternal, one from mom called the maternal. All right. Now, genes occupying the same position or locus on a homologous chromosome, these are known as alleles. And alleles occupy that same location. So you see here the locus for gene A, gene B, and gene C. You'll notice that we have two different alleles for gene A, and for both gene B and gene C, we have two of the same allele. So the maternal and paternal chromosome are matching for these last two gene locations. And any individual, barring some sort of chromosome number mutation, will always have two copies of any particular gene, meaning they can have a maximum of two alleles for any given gene. However, we'll talk more about this later, but there may be more than two alleles possible for a particular gene. A good example of this is blood type. You can have allele A, allele B, or allele O, but you can still only have two of them. You can't have all three because you only have two chromosomes, one from mom and one from dad. All right, so this is the other slide that's in the handout that I went ahead and attached to this post. We start out at the bottom with maternal and paternal chromosomes. Okay. Remember that when we go ahead and look at these together, they make up what's known as a homologous pair. One from mom, one from dad, one from the egg, one from the sperm. You'll notice that we have different colors highlighting the particular locations of a gene. Remember, a location is known as a locus or position. And for this particular trait, we have two of the same allele. But alleles, remember, are versions of a particular gene. If you have two different versions, like you see with gene A, one capital that's dominant, one lowercase that's recessive, this is what's known as heterozygous. That prefix hetero means different. Okay. In these particular instances, if we have traditional inheritance, you would see that the dominant trait will overshadow the recessive trait. If we go to gene B, you see that there are two of the same allele. This is what's said to be homozygous dominant, homo meaning same. And for this particular case, we'd call it dominant because they're both capitals, they're both dominant genes. For gene C, we'd call this homozygous recessive because, again, you have two of the same allele, but instead of them being dominant, you have two recessive alleles. Now, when you think about what you have in terms of your genetic combinations, you got some from mom and some from dad. So which one do you pass on to your offspring? The simple answer is it's pretty random, okay? You don't know exactly which allele, which gene is going to be passed on until you do so. You can talk about probability, but ultimately whatever one goes in is what you end up passing on. Now, did you give on your mom's or your dad's? Well, it could be either one. And it could be different depending on which chromosome it is. You could give chromosome 1 from dad, chromosome 2 from mom, chromosome 3 from mom, and so on and so forth. The passing on of these traits or these genes is because of what Mendel called the law of segregation. Okay, so this result of the way each allele is carried onto separate homologous chromosomes that separate during meiosis. So understanding meiosis is important here. It depends on which of the gametes you got at the end of meiosis. For any particular gene, remember an individual may be homozygous, two of the same allele, or heterozygous, two different alleles. But unless there's a chromosome number mutation, they will only have two alleles. Gametes contain only one copy of each, since you receive one chromosome from each parent, either in the sperm or the egg. So you begin with a homologous pair. Remember, this is what it looks like when we go ahead and get to metaphase 1, and then it will continue to divide anaphase 1 all the way through to telophase 2, and you will end up with 4 different gametes. Okay, we'll go over this concept of segregation a little bit more and independent assortment as well. These are two big things from Gregor Mendel that we need to highlight in a little more detail. So let's make sure you know how to do a Punnett Square. Now, the name actually comes from the British geneticist R. Punnett, who devised a method for calculating all the possible combinations of gametes and offspring. You see the little square down in the bottom. Essentially what happens is you take the original cross and you go ahead and you separate out the alleles. So if I go ahead and separate out the first parent, let's say this is the maternal, okay, and we go ahead and get one dominant P and one recessive P. That's P for purple. We typically like to use a letter related to the dominant trait. But then we go ahead and do the same thing for the other parent, big P and little p. Now, the important thing to make sure you can keep in mind is how do you distribute these alleles, okay? When we do so, you're going to go ahead and go down and down. You're going to go ahead and go across and across. So if you watch this and follow your fingers along, you're going to have a big P from here and a big P from here, and there's your big P, big P. You have a big P from here, a little p from here. We have big P, little p. Okay, and so this is how you're going to go ahead and fill out Punnett squares. Now, ultimately, you have to remember that the dominant trait, if present, is going to be expressed. So it's easy in big P, you're going to have purple flowers. But notice that when we have big P, little p, we also have purple flowers because the big P for purple dominated over the little p for white. But in situations like the bottom right-hand square where you have little p, you end up with white flowers because you got two recessive traits. Okay? All right. So I hope that helps kind of review and remind you of some of the stuff you went over in middle school. We'll just get plenty of practice with this. And so make sure you kind of just get the hang of this. And if there are any questions, please come see me. All right, guys. Thank you so much. I'll talk to you soon. Take care of yourselves. Bye-bye.